Namaskar. Now I am going to deliver a fourth lecture of cell cycle regulation regulatory pathway. Already in previous videos, I have discussed what are actually the different CDK proteins, cyclin proteins, how they regulate one another, and how they control the cell cycle progression through G1 to S phase, and why this point is very much important. What are the different CDKs? What are the tumor suppressor genes actively present and protect us from the disease, the dangerous disease cancer that I have already discussed. The few pathway of retinoblastoma, RB gene pathway, RB protein pathway, RB protein regulation, and P53. P53 protein is a vital protein, is known as a guardian of our genome, and if the RB protein is getting mutated, or if P53 protein is mutated, it will cause a cancer. More than 50% cancer is due to the mutation in the P53 gene. So what are actually the pathways? Today now I will discuss about RB pathway, that is retinoblastoma gene pathway, protein pathway, and P16, another stress response gene pathway, stress response protein pathway, and how the P53 protein is again involved in programmed cell death or apoptosis. Now let's see what happened in RB protein. The retinoblastoma protein is a tumor suppressor gene, the most vital tumor suppressor gene. It is a main common pathway protein. What happened if RB protein is okay, if RB protein's function is normal, then RB protein binds automatically with E2F transcription factor protein. E2F is present mainly in higher eukaryotes, in mammals and eukaryotic plants, and in mammals they are present, and these are actually a class or a family of transcription protein. They transcribe a transcription factor protein, that is E2F is a gene that gene produces a family of transcription factor proteins, and here I have mentioned the E2F regulators. This RB protein, when is normal, that is no mutation occur in RB protein, cell is okay, then what happens? RB protein binds with E2F protein, and E2F protein is non-functional in that condition. So E2F protein is not able to transfer from cytoplasm to nucleus, so that it will transcribe the genes responsible for the G1 to S phase transition. So RB protein is tumor suppressor protein. What happens if RB protein will be mutated? When RB protein will be mutated, then RB protein will not able to bind with E2F regulator protein, and E2F will be free. If E2F will be free, then it will not able to inhibit the gene transcription that are responsible for G1 to S transition, that is cell cycle will run continuously. E2F inhibition is essential for controlling the cell cycle or for stop at the G1S phase. The cell cycle stoppage is necessary, and this E2F protein is very important. One most important thing, two virus. One is human papilloma virus, HPV, the key virus which is responsible for the cervical cancer of humans. This human papilloma virus, if integrates into the human genome by accidentally, if they infect human genome, then it viral character is that its DNA will integrate it into our DNA, and virus will produce huge amount of E6 and E7 protein. Here is a small pathway, HPV, human papilloma virus, integrates into our body and produce E7 protein. This E7 protein directly inhibit RB protein, so that it will cause a cancer. When RB protein will be inhibited, E2F will be free, and when E2F will be free, it will continue transcription of G1S transition, that is cell cycle will run continuously. So this is very important virus. Another important virus is adenovirus. We know that the adenovirus will produce another protein, E1A. this is also an inhibitor of RB, a tumor suppressor gene. This will also cause a cancer. So viral type of cancer, actually virus integrates in our body. They produce their proteins, and their protein are strong, so strong that they strongly inhibit our own regulatory proteins, so that cell cycle continue and cause cancer. Now, in the second case, when stress occur, any stress or any kind of stress sensor, any kind of stress occur or reactive oxygen species develop in our cell, a stress sensor protein is there, P16. This P16 protein will be activated, and when P16 protein will be activated and normal, then this directly inhibits CDK4 by binding. CDK4 when inactivated, automatically RB phosphorylation will be hindered. RB will not be phosphorylated, and RB is CDK4, sorry, in this case RB protein will be unphosphorylated. CDK4 when hindered, RB protein will be unphosphorylated. When RB is unphosphorylated, it will bind with E2F protein. E2F, again, E2F is not free in that case, as E2F is bind to the RB, is not able to transfer from cytoplasm to nucleus, so that G1S transition is not possible. So this is a very important another suppressor, tumor suppressor gene or stress sensor gene. If this protein will be inhibited, what will happen? CDK4 will be active. When CDK4 will be active, it will be phosphorylated. When it will be phosphorylated, it will free the E2F protein. When E2F protein is free, it will activate, it will actively transcribe the G1S transition proteins. So this one is very important in that respect. Sorry. Sorry for disturbance. Now, what happened? The P16 stress response protein or stress response gene is normal. If normal, then whenever stress will come, it will protect us from G1S it will protect us at the G1S stage. So this is very vital protein, stress sensor protein. Now, what will happen? The P53, another our guardian of our genome is the P53. What happened? P53 not only protect us from various kinds of tumors, but also it causes a programmed cell death. Programmed cell death, that is apoptosis. Along with the time, cell division is necessary and dying of the cell is again necessary. What happened? P53 gene produces the P53 protein. If in normal condition, if P53 protein is not mutated, then P53 gene produces the P53 protein. Then P53 protein when active, it will inhibit the BCL2 gene. What is BCL2 gene? BCL2 is the B cell lymphoma 2 gene. This gene produces the BCL2 protein. The P53 protein inhibit the BCL2 protein, and BCL2 protein again inhibit the apoptosis. So BCL2 is not good in that case. BCL2 inhibit the apoptosis. That is, if BCL2 protein is active, then it will inhibit apoptosis. That is, inhibit apoptosis, the programmed cell death will not occur. That cell will not die. That's if cell will not die, then it will cause cancer. So P53 is essential. P53 what is doing? P53 directly inhibiting the BCL2. So apoptosis occurs. If P53 is getting mutated or any kind of this abnormality occurred in P53 protein, then BCL2 protein will be active and it will inhibit the apoptosis, our programmed cell death. That is, they may lead to the cancer. And what is actually the details function of BCL2? BCL2 is a B cell lymphoma protein mainly present in the outer surface of the mitochondria. Mitochondrial outer membrane they attach here. Mitochondrial outer membrane BCL2 present, and it inhibits the two protein, pro-apoptotic protein. Pro-apoptotic protein are the Bax and Bak. This protein helps release cytochrome from mitochondria to cytoplasm. What is actually the Bak and Bax? These two proteins are very important. These two proteins are very important as for apoptosis. If Bak and Bax is active, then they will release cytochrome. Cytochrome, we know in respiration, cytochrome C is present. Cytochrome C is present in the inner membrane of the mitochondria. Cytochrome is present in the inner membrane of the mitochondria. It's very important for electron transport from complex 3 to complex 4. So when cytochrome is released from the inner membrane, cytochrome C will bind to the Apaf. Apaf means apoptotic protease activating factor. Apaf. It binds with the Apaf and procaspase 9. These three together constitute, three musketeers together constitute the apoptosome and called programmed cell death. So what is actually the summary? If BCL2 is not present, if BCL2 is inhibited by our P53, that is a normal human being, P53 inhibit BCL2, then Bak and Bax helps to release cytochrome C, and cytochrome C from the inner mitochondrial membrane. Cytochrome C will bind to the Apaf1. Apaf1 along with the procaspase 9 will activate the apoptosome and program cell death. That is, a cell will die in a normal way. If P53 is mutated, BCL2 will be active. BCL2 when active, it inhibits the Bak and Bax protein, pro-apoptic protein. So why? Cytochrome C will not be released and cell will not die. Apoptosome is not formed. So this is very important, BCL2 protein and P53 relationship. In a later video we will discuss about apoptosis in details. Now here is actually the BCL2 pathway. So what I have discussed? I have discussed about RB protein function, RB protein pathway of cell cycle regulation, retinoblastoma gene. If single copy of RB will be mutated in a human, then no problem, he will carrier. But if two copy of RB will be mutated, both RB, then it will cause a cancer of retina. It's called retinoblastoma disease. And RB protein function, how RB protein regulate E2F, and E2F how regulate the G1S cycle transition, and how RB protein is regulated by two viruses, HPV and Adenovirus. What happens if RB protein will be mutated? Then how the cell will run continuously from G1 to S phase uninterruptedly? It will cause cancer. P63 protein, what is P63 protein and how stress response cause the direct inhibition of RB? So it will cause it will protect us from cancer in the same pathway as RB, and the P53 inhibit BCL2. So apoptosis occurs properly because BCL2 inhibit apoptosis by Bak and Bax inhibition. Bak and Bax, our friend, they release cytochrome when cell is needed. They release cytochrome from mitochondrial membrane to the cytoplasm so that it will attach with the Apaf1 and procaspase 9, and these three together form apoptosome. This apoptosome cause programmed cell death or apoptosis, which is most important whenever cell is damaged or diseased. Apoptosome formation is necessary for its destruction. If not occur, then cell will go for cancerous growth. Thank you.